Imagine being able to turn a specific thought, a physical movement, or even a deep memory on and off with a single flash of light. Now, that sounds distinctly like science fiction. But right now, inside animal models in laboratories around the world, this level of control is a daily reality. It is made possible by a biological technique called optogenetics. Put simply, optogenetics combines optics, the use of light, and genetics to directly control the electrical activity of the brain. Instead of guessing which of the brain's 86 billion neurons are responsible for a specific behavior, researchers can now isolate and program these individual circuits with exact precision. To understand how this works, we have to look at the basic hardware, your neurons. These cells are essentially microscopic electrical devices that communicate with each other by firing tiny impulses. For decades, scientists struggled to control these impulses precisely. The solution ended up coming from a completely unexpected place: simple green algae. These organisms contain naturally occurring light-sensitive proteins called channelrhodopsins. Using modern gene therapy, researchers can extract the instructions for these algae proteins and insert them into a highly specific, targeted cluster of neurons in a lab subject. Once those neurons produce the protein, they become photosensitive. When a highly specific wavelength of light, like a precise beam of blue light, hits the targeted neuron, the algae protein reacts. The reaction is entirely mechanical. The blue light forces the channelrhodopsin to change shape, prying open a microscopic ion channel on the cell's surface. That opening creates a doorway. Positive ions rush in, shifting the electrical charge and instantly triggering the neuron to fire. The process works in reverse, too. Researchers can use different variations of these proteins, called opsins, that react to different colors. Exposing them to an amber or yellow light will pump negatively charged ions into the cell, silencing the neuron instead of activating it. Historically, scientists had to use blunt electrodes that shocked a wide area of the brain all at once. Now, they can target and trigger individual cells with surgical precision, leaving the surrounding tissue completely untouched. Because light can be pulsed in fractions of a millisecond, researchers can match the incredibly fast processing speed of the brain. This lets them map out densely packed neural pathways one connection at a time. In mice, this technique allows scientists to map and control complex physical behaviors. By sending light through a fiber optic cable attached to the head, they can instantly alter its feeding habits or trigger specific mating behaviors on command. These applications reach far beyond just the physical, into the cognitive level. Scientists have used targeted light to trigger the recall of a specific fear memory in lab subjects, or even erase the response entirely. By isolating exactly which cells control these responses, these laboratory experiments prove that our complex behaviors, thoughts, and fears are tied to physical, manipulatable pathways. Applying this to living humans, however, introduces a massive physical hurdle. Human brain tissue is incredibly dense, meaning it easily absorbs and scatters light. If a target circuit is buried deep inside the brain, shining a light from the outside won't reach it. Getting the light to the cells requires implanting invasive hardware directly into the tissue, like fiber optic cables. That level of invasive surgery carries inherent risks, which currently prevents the widespread use of deep brain optogenetics in human patients. Engineering a safe, non-invasive way to deliver light through the skull and deep into dense tissue is the primary roadblock the field has to solve before these molecular therapies can reach everyday clinics. But researchers have found the eye. In a recent clinical trial, researchers treated a patient who was completely blind from a degenerative disease called retinitis pigmentosa. They injected an optogenetic gene therapy directly into the surviving but dormant cells of the patient's retina. To activate those cells, the patient was equipped with specialized engineered goggles. The goggles read the surrounding environment and pulse amber light into the eye. The treated retinal cells detect those light pulses, fire their electrical signals down the optic nerve, and the patient was able to partially regain their vision. That visual recovery is just the beginning. Major funding groups like the NIH Brain Initiative are aggressively funding this research, aiming to build precision therapies that can safely reach deeper into the brain to correct circuits responsible for Parkinson's disease, severe depression, and other neurological conditions. A microscopic light switch, borrowed from a simple alga, is now illuminating the future of human medicine.